In this video today, I like to present you medicinal chemistry of neurotransmitters. And also during this video, I like to give you the big important points that are always expected in the exams. Let's start. First, let's see what is neurotransmitters. The neurotransmitters are chemicals that communicate between two neurons or nerve terminals. And these neurotransmitters are biosynthesized in the presynaptic neurons and then diffuse into the synapse, and then they bind to the receptors in the postsynaptic neurons. And once they bind to those receptors, they produce signals, and that helps in various physiological activities. So these neurotransmitters are several types. In this video, I like to focus on the two major types of neurotransmitters. Those are monoamine types, and then the second ones are amino acid types. So monoamine types are further divided into quaternary amines, indole amine types, and then catecholamine types. And monoamines, quaternary amines, the best examples are acetylcholine. Acetylcholine is a cholinergic neurotransmitters. And then indole amines, the best example of indole amine type of neurotransmitter is serotonin. However, the next catecholamine type of neurotransmitters are very well-known transmitters that are for sympathetic or adrenergic system. Among the catecholamines, the mainly dopamines, norepinephrine, and then epinephrine. These three types of neurotransmitters, also known as catecholamine neurotransmitters. And then next, amino acid type of neurotransmitters. In the amino acid type of neurotransmitters, primarily we discussed today about glutamate, GABA, which is also known as gamma aminobutyric acid, GABA aminobutyric acid, and then glycine, and then serine. So let's start first with monoamine type of neurotransmitters. That is acetylcholine. Acetylcholine is synthesized in presynaptic neurons. So let's discuss this entire process in two simple steps. The first step, how they are synthesized and where they're released, and also what kind of receptor they bind to them. And then after the second step, after formation and binding to those receptors, these signals will terminate. Also, we'll look at what are those enzymes that is necessary for those termination of those signals. So first let's see the biosynthesis of neurotransmitter acetylcholine takes place on the presynaptic terminals, and then there are postsynaptic terminals, there are receptors, and they bind to them and they produce physiological action. So here, let's talk about how this acetylcholine is synthesized or biosynthesized. So choline enters into the presynaptic terminal by the choline carriers, and this choline in presynaptic terminal undergoes biochemical reaction by enzyme called choline acetyltransferase. This choline acetyltransferase produces the acetylcholine, and this acetylcholine released into the synapse, and then eventually those acetylcholine bind to the acetylcholine receptors in the postsynaptic terminal. And after this binding to them, there is the excessive neurotransmitters, acetylcholine undergoes signal termination. And signal termination takes place by the enzyme called acetylcholinesterase enzyme. And these acetylcholinesterase enzymes break down acetylcholine to acetyl and choline group. And this acetylcholine, here you have a question about the question alert on acetylcholine. Acetylcholine signals are terminated by the enzyme. What enzymes terminate them? The answer would be, guess what could be the answer that could be expected in the exam. So because acetylcholinesterase enzyme plays very important role in terminating, that means acetylcholine breakdowns to acetyl and choline by acetylcholinesterase enzymes. We will see those are the cholinergic agonist drugs, or primarily they act on by inhibiting acetylcholinesterase. By doing so, you have higher acetylcholine levels in synapse. Let's look at what are those cholinergic receptors these acetylcholine bind to them. Acetylcholine, which is a cholinergic neurotransmitter, or also known as parasympathetic neurotransmitter, act on or bind to the two types of cholinergic receptors. One, muscarinic, and then other one, the second type is nicotinic receptors. And within muscarinic receptors, there are 5 types of muscarinic receptors: M1, M2, M3, M4, and M5 receptors. Among those, however, the distribution of M3 receptors is more popular, which has distributed various parts of the body. However, let's see what exactly M1 receptors are. M1 receptors are mainly found in the CNS and primarily plays important role in cognitive functions. And then M2 receptors are mainly present on myocytes, and that helps in contraction of the heart cells. And the M3 are more likely potential for the exams. And most drugs, they do reverse the effect of M3 receptors because M3s are distributed various organs, especially peripheral smooth muscles contractions. Like they are primarily present in the bladder, eyes, and also the gastrointestinal tracts. So therefore, they play a vital role in cholinergic system. And then M4 receptors are CNS and they modulates neurotransmitter release, and M5 also present mainly in CNS. They control the movements or motor neurons. The next type of receptors for cholinergic neurotransmitter acetylcholine are nicotinic type of receptors. Within nicotinic type, there are 2 types of nicotinic receptors. One is a NM, also known as NMJ, and then second type is NN type of receptors. And NM type of receptors, or which is present mainly in the neuromuscular junction, and that helps in depolarization, and that helps in muscle contraction. So most famous drugs such as NMJ blockers, like succinylcholine or pancuroniums, those drugs, they act on those NMJ junctions by acting through the NM receptors. And NN are type of neuronal type of receptors mainly found in central nervous system and peripheral nervous system. And NN further divided into alpha and beta. Not to be mistaken, not to get confused, these alpha and beta are not the same type of sympathetic receptor. They are different type of receptors, which is primarily present on in the neuronal type of central nervous system cells. So there are various, within this alpha, again, you have alpha 1, 2, 3, 4, and beta 1, 2, etc. There is one important drug that acts through the alpha 4 and beta 2, that is a vernaculine, or the brand name is Champex or Chantex. Those drugs, they act through alpha 4 and beta 2 receptors. So what are those takeaway points for cholinergic receptors? Or keep eye on those M3, that's a big in the exams, and of course nicotinic, NMJ blockers, and then neuronal type NN, which is the alpha 4 and beta 2 receptors. Next, let's talk about the distribution of catecholamines in the brain. The catecholamines are type of neurotransmitters that are known for sympathetic or adrenergic system activities. And within this catecholamine, there are three major catecholamines plays important role as a neurotransmitters. One is a norepinephrine, epinephrine, and then dopamine. So the distribution of norepinephrine is primarily present in the brain. In the brain locus coeruleus, site where you will find more norepinephrine is synthesized. Epinephrine is not primarily present in the brain, but there is a small amount of epinephrine also present in the brain that is part of the medullary epinephrine neurons. However, the major catecholamine that is present in the brain is dopaminergic neurons. They produce dopamine, which is primarily produced from substantia nigra and ventral tegmental areas. Now let's talk about these catecholamines chemistry. What exactly this medicinal chemistry of catecholamine is about? Because catecholamines are major neurotransmitter of sympathetic or adrenergic system. So here the structure of dopamine. So dopamine is a good example of catecholamine. And this dopamine catecholamine means catecholamines are characteristic. Structurally, they do have a two hydroxyl group next to each other. That's what makes them as a catecholamine. And then you have a ethylamine onto this benzene ring. That's a very characteristic structure for dopamine. Dopamine structure is always important for the exams, so they can ask you direct question on examples of dopamine structure. So here further, also you have to know few more points on this dopamine structure because dopamine is very important characteristic structure for neurotransmitters such as sympathetic neurotransmitters. So dopamine, you can designate this a carbon next to the nitrogen is alpha carbon, and then here you have a beta carbons. So now this dopamine undergoes biosynthetic reaction, which is by dopamine hydroxylase. And in this dopamine hydroxylase means in this beta position, you will find a hydroxyl group. So formation of beta position hydroxyl group, therefore it is called dopamine beta hydroxylase. And this structure is for norepinephrine. So you could see very structural similarity between norepinephrine and dopamine, just one extra group that is attached that is called hydroxyl. And therefore this enzyme is beta hydroxylase. So easy to remember the names of the enzymes. Sometimes the questions also can come in the exam about the enzymes. So the enzymes of this are always the end with the ase. So since you're adding hydroxyl, so since therefore it is called beta hydroxylase. Then further, norepinephrine undergoes another reaction by N-methyltransferase. That means you will find a methyl group on top of nitrogen. So when you attach methyl group, that is called N-methyltransferase. And this structure is for epinephrine. So these are the three major catecholamines. However, norepinephrine, epinephrines are primarily plays important role in sympathetic neurotransmission. Next, let's look at the norepinephrine. Well, norepinephrine produced here, as I said before, here you got presynaptic terminal and then postsynaptic terminals. So in simple, I can present this process, entire process of this in two simple steps. One is formation of neurotransmitters and then binding to the receptors. And then the second step is all the neurotransmitters that terminate, the signals are terminated and termination process by the enzymes. So first step is how this norepinephrine is synthesized. And the second step, I'll explain you what are those enzymes responsible for termination of signal. Because after this neurotransmission and binding to those receptors, signals are terminated by various enzymes. So first let's look at norepinephrine. Norepinephrine is mainly synthesized from amino acid, that is a tyramine. Tyramine in the presynaptic terminal undergoes metabolic reactions such as it makes the L-DOPA. Tyramine to L-DOPA is done by decarboxylation. L-DOPA stands for levodopa. This levodopa becomes dopamine, and this dopamine structure we have seen before in the previous slide. This dopamine breakdowns to norepinephrine by what enzyme it does? It does hydroxylation. Beta dopamine beta hydroxylase enzyme. This dopamine beta hydroxylase enzyme makes norepinephrine in presynaptic terminal. And this norepinephrine vesicles will diffuse into the synapse and they release into the synapse and they bind to the receptors. And those receptors produce various physiological activities. And then after that, the remaining norepinephrine undergoes termination. These signals are terminated by various metabolic enzyme reactions. So there are two major enzymes plays important role in breaking down of the norepinephrine or terminating norepinephrine signals. One is MAO, which is a monoamine oxidase, and that terminate those norepinephrine. And then also another enzyme called COMT, which stands for Catecholamine methyltransferase. These two enzymes plays important role in terminating norepinephrine signals. Let's look at those chemistry of those two types of enzyme reactions, which is a monoamine oxidase, which is a MAO. MAO stands for monoamine oxidase. There are two types of MAO enzymes, MAO A and MAO B. And the second enzyme that plays important role in termination is catechol O methyltransferase. So first here, let's take an example of a dopamine, how this dopamine undergoes this termination by these two enzymes. First here, let's talk about MAO enzyme. MAO does an important reaction that's a very frequent questions in the exam. The big in the exam is the name of that MAO reaction that is known as oxidative deamination. Oxidative deamination is a big for the exam. So this oxidative deamination breakdowns MAO to MAO enzyme, catalyze that reaction and make this amine group, and then amine is eliminated, deamination, and then this carbon become oxidized to the acid. So this transformation is known as oxidative deamination. And the second enzyme, that is catecholamine methyltransferase, COMT enzyme, that, as the name tells you, the name is methyltransferase. That means, imagine, guess what we expected here? You expect one methyl group here. This methyl group formed onto this dopamine. So that is how the catecholamine methyltransferase metabolize those catecholamines. And then further, this also undergoes methyltransferase, produce this one, and then this also undergoes MAO oxidation, oxidative deamination. So this process of metabolic reaction terminate those catecholamine type of neurotransmitters. So this process applies to all three monoamine types, such as dopamine, norepinephrine, and also epinephrine. Next, let's see adrenergic receptors or sympathetic receptors. So here, I like to take example of norepinephrine neurotransmitter and also what type of receptor it binds with. So norepinephrine neurotransmitters and epinephrine, both of them, they are primarily bind to the sympathetic receptors, which are divided into alpha and beta. There are two types of receptors. And further, alpha divided into alpha 1 and alpha 2 receptors. And beta further divided into beta 1, beta 2, and beta 3 receptors. So alpha 1 receptors are mainly, they are known to activate blood vessels or vascular system, and they are known to cause strong vasoconstriction. So the big point about this is, the huge in the exam is alpha 1s are strong vascular constrictors, whereas the alpha 2s primarily present in the brain or central nervous system, and they do have negative feedback into the norepinephrine. That means when you activate the alpha 2, they reduce norepinephrine activity. So they mean they inhibit the norepinephrine release. So that's a very unique negative feedback that happens through the alpha 2 receptors. Whereas beta 1 receptors, they are primarily present in the beta 1. Like there's a mnemonic for one for heart, beta 1 for heart. That means they are mainly present in the heart. In the heart, they increase heart rate, force of contraction, and that leads to the increase in cardiac output. Eventually, that increases the blood pressure. And then next, beta 2s are mainly present in vascular and also bronchodilation, lungs, so they lead to the bronchodilation. Whereas beta 3 is not that common, but the one important site beta 3 is popular, that is in the bladder, detrusor muscles, helps in muscle relaxation. That means it helps in bladder filling. So therefore, filling the bladder is done by detrusor muscles, which are beta 3 receptor, and there's only one drug currently available that acting through the beta 3 receptors, and therefore that's not common, but that can be important for the exam as well. All right, so here is a question alert. So sympathetic stimulation of alpha receptor causes what? Guess what is the best response for this question alert here? Yes, you guess and answer this into the description in our channel, and then test your answer. So answer here for this one is, when you stimulate the sympathetic alpha 1 receptor, that causes vasoconstrictions, and not the vasodilation. That's wrong. Here, corrected this one, vasoconstriction, because sympathomimetic activity causes vasoconstriction, whereas alpha 1 receptor blockade causes the vasodilation. All right, the next one is dopamine receptors. So dopamine is dopamine neurotransmitters. We have seen the structure, which is a type of catecholamine neurotransmitter. And this dopamine bind to the 5 dopamine receptors, D1, D1, D2, D3, D4, and D5. However, D1 and D5 have a common sites, and they commonly mainly found in central nervous system, and they do play important role in excitatory, cognitive, and motor and reward functions. And D2 is a big for the exams. D2 and D3 have something common. They do both are G protein coupled receptors, and then they're primarily found in central nervous system, and they have inhibitory activities, and then they do have effect on mood, emotion, and motor function. So keep eye on the D2. D2 can be a big deal for the exam. And then finally, you have a D4. D4 are, they also G protein coupled receptors, and they are primarily found in central nervous system, plays important role in attention and impulse control. That means that sounds like is attention deficit hyperactivity related activities, or D4 receptors. Whereas D2 is a big, because D2 plays important role in motor functions, emotions, and mood. So here is a question. The famous drugs that are antipsychotic drugs alleviate schizophrenia symptoms by what? Symptoms by inhibiting what T receptors, dopamine receptors. Guess what could be the answer? The best answer would be D2 receptors. Inhibiting D2 receptors, antipsychotic drugs, helps in reducing schizophrenic symptoms. All right, so let's look at the next not the catecholamine type, but it is a indolamine type of neurotransmitter. that is known as serotonin. Serotonin is a big in many drugs. They act through the many class of drugs, plays very important role by action through serotonin neurotransmission. So let's look at how these serotonins are synthesized and produce their activities. Obviously, serotonin is synthesized from in the presynaptic terminals, and then once it is diffused into synapse and then bind to the receptors, most of them on postsynaptic terminal. And also, and so here, serotonin is produced from amino acid that is known as tryptophan. Tryptophan makes serotonin, which is simple structure, and this serotonin is diffused into the synapse and then bind to the serotonin receptors. And these serotonin receptors produce various physiological activities. And then the rest of the serotonin will undergoes deactivation or termination by reuptake mechanism. There are 5-HT reuptake transporters present on the presynaptic terminal, and they take up back those serotonin neurotransmitter back into the presynaptic, and they recycle back into the in the serotonins. So that is the termination mechanism of serotonin. Next, let's look at the types of receptors. That is, um, before we go to receptors, first let's look at the medicinal chemistry of this indolamine. So just to remember, just to let you know that indolamine, unique thing about it is the ring. The name of this ring is called indole ring. That's what you get this indolamine name come from, right? So this indolamine then further undergoes. First step is you see, notice here what happening here, hydroxyl group. That means tryptophan hydroxylase. This hydroxyl group activated by tryptophan hydroxylase and then intermediate product and then final product would be serotonin, which is eliminating carbon dioxide. So by removing carbon dioxide, it becomes 5-hydroxytryptamine. In short, it is also known as 5-HT or serotonin. This sequence is very important. Often you expect question on the sequence of formation of this serotonin. Now let's here see the question. What kind of question can be expected from here? What are those two enzymes, enzymatic reaction that helps to produce tryptophan to serotonin? First one is a hydroxylase and the second one is decarboxylate. That's your answer. All right, let's look at the serotonin receptors. There are serotonin bind to the various receptors. Here I'm only presenting the major ones. There are three major receptors, serotonin neurotransmitter bind, 5-HT1, 5-HT2, and 5-HT3 receptors. Again, 5-HT1 has a 1A, 1B, and 1D receptors. And especially 1A is a big popular. 1A is primarily plays important role in mood and anxiety regulations. Many antidepressant and anti-anxiety drugs, they act through 5-HT1A receptors. Whereas 1B found in the brain plays more important role in motor functions and also especially pain modulations and vasoconstriction. So therefore, drugs like triptans or a drug that are used for migraines, etc., they do act through those receptors. However, 5-HT2 plays very important role in various psychiatric functions, such as 5-HT2A plays important role in schizophrenia because it does associate with the hallucination. And 5-HT3 especially found in the GI and also present in the nausea vomiting centers. So therefore, most important antiemetic drugs such as cetrans, they act through the 5-HT3 receptors. So simple question here, what is the name of the ring that is present in serotonin? Here is a question. Guess what could be the answer for this serotonins? The name of the ring is indole ring. Indole ring is present in serotonin. All right, so let's move on to the next type of neurotransmitters, such as amino acid neurotransmitters. In amino acid neurotransmitter, there are mainly three major neurotransmitters here we're discussing. One is glutamate. Glutamate plays very important role. Glutamate further undergoes decarboxylation to form another neurotransmitter that is known as gamma aminobutyric acid. In short, this is known as GABA, which is known for famous GABA. And then this serine, another amino acid, produces the glycine, another amino acid, by serine hydroxymethyl transferase. Let's talk about glutamates. Glutamates are very important neurotransmitter, which is a very often expected in the exam from the glutamate. So glutamate, how they are biosynthesized. Glutamates, obviously, as I said, the same synthetic process, such as first they are produced from biosynthesized in the presynaptic terminal, and then they diffuse into the synapse, and they bind to the receptors. And the second important process is they terminate the signal by reuptake process. So here, let's look into this glutamates. Glutamates are produced from glutamine amino acid, and then here glutamines also produced from uh, glial cells and also presynaptic. And there is an enzyme glutaminase, and that enzyme activates glutamine to glutamate. Just a simple difference here. Structurally, here you have a ester acid, here you have a amine. So in this one, now once their forms into the presynaptic vesicles, those diffuse into the synapse and they release neurotransmitters here. And glutamates are bind to two major receptors. One is called NMDA receptors and then AMPA receptors. And then eventually that produces the physiological activity of glutamates. And then these remaining signals will terminate by going up into the glial cells and then also into the presynaptic neurons. All right, so let's look at the examples of glutamate receptors. The two major glutamate receptor, NMDA, and these are very big receptors because all those glutamate receptor NMDA are ion channel type of receptors. They primarily helps in calcium, sodium, potassium ion entry into the cell. That plays a very important role. And these are excitatory receptors. And then they play important role in memory and learning or cognitive functions. Then you have AMPA receptors, AMPA receptors also plays ion channel. They help in calcium and sodium, potassium ion entry into the cell. And then the three type is kinase type of receptors. They do not very have studied much, but first NMDA receptors are big for the exam, so you should keep eye on them. And there are, other than these three, are ion types, and this also metabotropic or the G protein couple receptors, which is they call glutamate receptor, also been recently investigated. However, keep eye on the NMDA receptors. So here, which glutamate receptors are primarily involved in cognition process? Guess which are those plays important roles of glutamate receptor that is often used also in dementia or Alzheimer's dementia treatment as well, which is NMDA receptors plays important role in cognition. All right, so let's look at the next GABA. GABA is known for GABA inhibitory neurotransmitter primarily found in the brain. And this GABA also biosynthesized in the presynaptic terminal, eventually diffuse into the synapse, and then bind to the receptors in the postsynaptic terminal, and then terminate through, signals are terminated through by going into the glial cells and also reuptake. So let's see how this GABA is synthesized. GABA is synthesized from glutamic acid. This glutamic acid decarboxylase, just removal of carboxylic group, produces GABA, which is a gamma aminobutyric acid. Once it is formed in these vesicles, presynaptic vesicles, release into the synapse and bind to the GABA receptors. And GABA receptors are well-known inhibitory receptors. And then further, termination of signal happens through entering into the GABA signals. So here is a question alert. What is the major inhibitor in neurotransmitter in the brain? That's the easiest question and one of the most frequently asked questions throughout the university's level exams and also, of course, board exams. So what is the best answer here? The best answer is GABA, which is a major inhibitor in neurotransmitter. So next, let's quickly look at what are those GABA receptors. There are GABA bind to the two major receptors, GABA A and GABA B. And GABA A are ligand-gated chloride ion channels. The big drugs, the big class of drugs, they act on them, that is benzodiazepines and barbiturates. Benzo and barbiturates are the big ones. They act through the GABA A receptors. And also, of course, alcohol also activated by GABA A. So, and therefore, the big class of drugs are here. So keep eye on the GABA A, that is more potential for the exam. Whereas GABA B is a metabotropic receptor, that means that is a mainly activated by the type of receptors or G protein coupled receptors or kinase type of receptors. So those are leads to the reduction in the neuronal excitability. The big famous drug here, baclofen, which is a muscle relaxant, acted through by GABA P. All right, so here is a question alerts. What are those benzodiazepine binds with the GABA A, which leads to what? What kind of effect it happens? Does it uh, that causes the induction of the GABA A activity or inhibition activity? So basically, benzodiazepine enhance the GABA inhibitory effect because GABA is an inhibitory neurotransmitter. That inhibitory effect is enhanced by the benzodiazepine and barbiturates, and that lead to decrease in chloride ion entry into the cell, and that helps in those benzodiazepinic effects. All right, so finally, we have a last neurotransmitter for today's presentation, the glycine. Glycine is also produced in presynaptic neurons. So here you have presynaptic terminal, and then obviously they diffuse into the synapse and bind to the receptors. So glycine is produced from amino acid that is known as serine. Serine hydroxyl methyltransferase produces the glycine, and this glycine are released into the uh, synapse and they bind to the glycine receptors, and then signals are terminated by getting those some of the glycine transporters take back into the presynaptic, and then also glycine will be entering into the glial cells. That's a termination mechanism. And these glycine receptors are do plays very important role in various physiological functions. Let's look at what are those. So here glycine receptors are basically there are two types of receptors. One is a glycine R and NMDA. So the most important point here is NMDA. We saw these NMDA receptors in previous glutamate. The same NMDA receptors also activated by glycine transporter or glycine neurotransmitter. This is a glycine neurotransmitter. And then this are keep eye on the NMDA, which is also act as an agonist with glutamate. So same neuroreceptors are bind with the glycine and also with glutamate. So here is the last question for you, question alert. NMDA receptors are activated by what? There are two neurotransmitter activate NMDA receptor. One is glutamate and then other one is a glycine. With this information, thank you for watching and hope you like this and find useful. And if you find this useful and you have many more videos are upcoming, so subscribe this channel and then give a big thumbs up. Thank you again.